Hello everybody and welcome back to Mr. Saucedo's YouTube videos. Today we're going to be going over lesson number 4, which is all about RNA transcription. So we've talked about RNA off and on in this unit, like we've mentioned it several times, but we've never actually defined what it is or really gone into detail as to what it does. And so RNA is actually super important, and as a result, we're going to kind of talk about what it is and then what it's used for today. So first things first, right? We have replication, which we talked about in the previous lesson. Transcription is used to make RNA from DNA. So replication is making a copy of your DNA. Transcription is using your DNA to make RNA. And you might think to yourself, why would you want to make RNA? What is RNA? So remember, DNA is trapped in the nucleus. So if you are the nucleus, the control center of the cell, and you need to order organelles to do things, the cell membrane to do something for you, to allow something in or to remove something, how do you send orders? So the way I think of it is, what if you were trapped in prison, right? If you're in prison, how in the heck are you going to be able to send orders, right? I mean, the nucleus is there for a reason. It wants to protect your DNA. So it can't just dissolve the nuclear envelope every single time it wants to send an order. That's not going to be very energetically efficient. Okay, so send a message, pass a note, right? So what it does, the nucleus, is it sends a messenger, and that messenger is called RNA. And the kind of interesting thing about RNA is it's very similar to DNA, but it does have some differences to it. So first things first, right? RNA stands for ribonucleic acid. So in case you forgot, remember the D in DNA stood for deoxyribose? Well, now we're just talking about ribonucleic acid, so it's not the same. And so in addition to that, there are 3 differences other than just the name. Number 1, it's single-stranded. No double helix here. In addition to that, oh, here you go. It's like a broken fence. Number 2, it's ribose. It's ribonucleic acid, not deoxyribonucleic acid. Okay. So look at the difference between deoxyribose and ribose. Very, very similar. Here we have a hydrogen. Here we have an OH. So not very different, but still a difference. And then the last one, which is probably the most important. RNA does not have thymine. So DNA has A, T, C, and G. RNA has A, U, C, and G. And the U stands for uracil. And so if you're looking at it, right, here's our adenine, here's our uracil, here's our cytosine, here's our guanine. There's no T, so no thymine, in RNA. So here is what a typical RNA molecule will look like. It is single-stranded. You notice you don't see any T's, but you do see U's. Okay. So remember, uracil is used instead. So A pairs with U, C pairs with G in RNA. Okay. There are 3 types of RNA, but they're all used in a very similar way. Okay. They all kind of fulfill different roles here. So the first type is the messenger that we were just talking about, right? That's called mRNA. The M stands for messenger, and it contains the blueprint for building proteins. So if you're the nucleus and you need something made, you're going to need a blueprint, and mRNA is what you're going to be making. So again, I always try to think of it like a foreman or something, right? Someone who has the blueprint, is aware of what's supposed to happen. Okay. Next is rRNA. Now, rRNA stands for ribosomal DNA. They actually build the proteins. And maybe you remember this, maybe you don't. So they're our construction workers. They are ribosomes. Ribosomes are made of RNA. We just never mentioned that when we were talking about cell organelles because it wasn't that important at the time. But ribosomes are made of RNA. That's actually what they're made of. And then tRNA or transfer RNA, those are like the tools that the construction workers are using. They transfer amino acids so that you can build your protein. Okay. How does transcription work? Thankfully, it's a lot simpler than replication. So the very first thing that happens, the nucleus needs a protein to be built. That's like step one. An enzyme called RNA polymerase is activated. Okay, so we have DNA polymerase, that is for DNA replication. We have RNA polymerase, that is for transcription. RNA polymerase attaches to the DNA in a specific spot, unwinds and unzips the DNA. No helicase needed. RNA polymerase does it all. And so here we have our separated now strand of DNA. So this is like, one half. This is the other half. And then this kind of bluish, teal, green thing, this is RNA polymerase. This kind of, I'm going to say orangey pink thing, this is our strand of DNA. And then this reddish pink stuff here, that is the RNA secret message, right? The messenger RNA that's being made. So next up, how does the RNA polymerase know to attach, right? Like, how does it know where to go? Well, it is on the lookout for a sequence in the DNA, and that is called a promoter. It signals the RNA polymerase, here, please attach here. This is an example of a promoter. It's one of the most common. It's called a TATA box. It's just TATA. And so RNA polymerase knows, okay, when I see TATA, I attach here. RNA polymerase creates a complementary strand. There's that word again, complementary. And it only makes that strand after the promoter. So this TATA box, this TATA, is not included. But remember, U is used in place of T because we're talking about RNA. So after this TATA, TATA, A goes with U. So that's what I would pair this up with. C goes with G. A goes with U. G goes with C. A goes with U. So this would be my messenger RNA that I'm making. How does it know where to stop? Once it hits what is called a terminator, it detaches, and now the transcription is complete. And terminators can be, again, a lot of things, but they're going to have to tell you. So like in a worksheet, on a test, in a problem, they're going to have to tell you what the terminator and promoters are because there are a bunch of them. So here we have half of a strand of DNA, and I want you to make the mRNA for it. It tells us the promoter is AC, the terminator is TC. So that means we read our strand from left to right, just like we would in English. And wherever we see AC, that is going to be our promoter. And that's wherever we see AC first. So it could be that this strand has hundreds and hundreds and hundreds of pairs, right? So it's got hundreds of different nucleotides. But you read it from left to right, and the very first time you see A and C, in this case, because it says so, that's going to be our promoter. And so I always like to say, put a box around it so that you know, okay, this was my promoter. What comes after that? We just start building our RNA. G goes with, C. That's not that different. A goes with, though, U. T goes with A. G goes with C, A goes with U, right? And you're now building it. And then once you get to your terminator, which I also put a box around, you stop. So this is our secret message, this blue thing right here. And this is in your notes, by the way. So in that box at the bottom of the page, make sure you fill this in. We're going to do a second one. See if you can do it on your own. Again, we've got a promoter, TA, terminator, CG. So I always say put a box around it, put a box around your terminator. Notice this isn't even going to be coded for anything. If the promoter is TA and the terminator is CG, the very first time you see a CG, that's where we stop here. So now let's keep going. A goes with U. Remember, it's RNA. T goes with A. And we have a lot of A's and a lot of G's. This is our RNA. All right, now let's see if we can go backwards. What if we were given the mRNA sequence? This is called reverse transcription. So we're trying to make the original piece of DNA from it, okay? And then it says to add this promoter and add this terminator. All right, so U pairs with what? A in DNA. A pairs with what in DNA? T. A pairs with, again, T, and then G, and then C. And so now I have my original strand of DNA, but it says to add our promoter and our terminator. So I'm going to add my promoter AC, add my terminator TC, and now technically I have my original strand of DNA that coded for this piece of mRNA. All right, that's it. So if you have any questions, please let me know.